The neurogenesis mechanism of desire involves the coordinated activity of multiple brain regions, whose operational patterns can be precisely observed through modern neuroimaging techniques. The entire process begins with the perception of environmental stimuli. The ventral tegmental area, serving as the primary hub for dopaminergic neurons, responds first to internal and external stimuli. These neurons project to the nucleus accumbens, forming the mesolimbic pathway that drives seeking behavior. fMRI studies reveal that when exposed to sexually related stimuli, the activation level of this pathway positively correlates with the intensity of subjective desire. The hypothalamus functions as an integrative center. The medial preoptic area receives olfactory signals, pheromones, while the arcuate nucleus regulates gonadotropin release. Together, these nuclei form a neuroendocrine conversion station, transforming physiological states into neural signals. The prefrontal cortex implements cognitive regulation. The dorsolateral prefrontal cortex assesses situational appropriateness, while the ventromedial prefrontal cortex processes value judgments. Enhanced activity in these regions often exerts inhibitory effects on the limbic system, reflecting rational control over instinct. The insula integrates interoceptive awareness, that this brain region maps bodily states into subjective sensations, interpreting physiological changes like accelerated heartbeat or skin warmth as signals of arousal. Anterior insular activity correlates strongly with desire experiences, reaching a correlation coefficient of 0.72. The amygdala performs emotional labeling. Through associative learning mechanisms, it links specific stimuli to pleasurable experiences. Once this classical conditioning is established, the associated stimulus can independently trigger desire responses. Neurotransmitters follow precise spatiotemporal release sequences. Dopamine dominates the anticipation phase. Norepinephrine heightens alertness, and endorphins generate pleasure during the experience's latter stages. Oxytocin promotes post-intimacy satisfaction and emotional bonding. Understanding these mechanisms holds practical significance. Reinforce amygdala connections through repeated positive experiences. Activate the dopamine system with novel stimuli, and reduce excessive prefrontal inhibition via relaxation training. Neuroplasticity confirms these methods effectively optimize desire generation pathways.